All right, everyone. Thank you for coming. Hope you're having a wonderful GTC. My name is Rory Kelleher. I lead global business development for life sciences here at NVIDIA, and I have the wonderful opportunity of introducing our panel for foundation models in biology. Just a few housekeeping things before we get started. Don't forget to download the NVIDIA GTC mobile app. It is super useful for finding your way around the zoo. Also, don't forget to explore the exhibit hall on level 2, just actually right outside here. It opens at 12 o'clock today, and there's also a reception between 5 and 7 p.m. tonight. Make sure you check out GTC Park, awesome food trucks, wonderful music, and just a great atmosphere, great vibes. And then the session recording for today and many other sessions will be made available on demand to attendees within the next 48 hours on NVIDIA On Demand next week. Awesome. All right, so I'm going to introduce our moderator here today. Anthony and I have known each other probably for 7 or 8 years, actually, when he was at Mount Sinai Health Systems. And he joined us because we needed somebody who really operated at the intersection of both artificial intelligence, biology, and at the time, he was also doing a lot in the clinical space as well. Today, Anthony actually leads digital biology, both products, so what we're building in BioNeMo and Parabricks and many of the other foundational libraries that NVIDIA is building for this space. And he also leads our developer relations team, so a lot of the subject matter experts who are engaging with companies like what we're going to hear from today, and I'm sure many of you in the audience. So with that said, please give a warm welcome to Anthony, who will introduce our panelists here as well. Thank you, Anthony.
Thank you. Right on time. All right, so thank you all so much for being here. It's really a pleasure. We have an incredible panel that I'm super excited about, and I think you'll all see why as we get through the hour that we have here today. So I'm going to introduce the panelists, and then we'll get going. So first on my left, we have Molly Gibson. Molly is an origination partner at Flagship Pioneering, where she leads venture creation team, founding and growing companies at the intersection of AI and science. She's the founding president of Lila Sciences, which are building new AI models to power the scientific method, and has been co-founder of Generate Biomedicines, where she served in key leadership roles, developing foundational technology and driving their overall strategy and vision. Her work has resulted in many patents and publications in science and nature, as you'll see, a common theme through our entire panel. Really accomplished people here, and been recognized in Endpoint's News 20 under 40 and in Business Insider's top biotech entrepreneurs list. Molly holds a PhD in computational and systems biology from Washington University in St. Louis, so welcome.
Thank you.
So Patrick Hsu is a co-founder of the Arc Institute and assistant professor of bioengineering and the Dev Faculty Fellow at the University of California, Berkeley. He's a pioneer in the field of CRISPR gene editing. His research group works at the intersection of synthetic biology, genetics, machine learning to invent new technologies to improve human health. He received his AM and PhD degrees from Harvard, and his research has been recognized by MIT Technology Review's Innovators Under 35, Vox Future Perfect 50, the Rainwater Prize, the Amgen Young Investigator Award, and among many papers that probably many of you in the field have heard of recently, including Evo2, which we'll talk a lot about here today. Nicholas Safroniev leads the Frontier Intelligence team at EvolutionaryScale, collaborating with scientists to apply advanced models to complex biological problems. Previously, he was director of product and technology at CZI, where he started many of the AI efforts in cell biology and imaging. And then last but not least, we have Joshua Meyer, co-founder and CEO of Chai Discovery, building AI foundation models for drug discovery. Prior to starting at Chai, he was chief AI officer at Absci and helped launch the protein folding division at Meta, and was one of the really OGs in developing the early parts of this field with early work on ESM. So we'll talk about that later today as well. So he was the co-lead author on many of the first transformer LLMs for proteins, and actually built MIT's CRISPR-ML design platform. Joshua completed his undergraduate and graduate work in computer science, also at Harvard. So welcome to this really incredible panel, and thank you all for being here. All right, so I want to get going with and talking about just defining what biological foundation models really even mean. And so if you look across this panel, we actually have an incredible diversity of the contributions to the field. We have work, like I mentioned, and introducing Joshua, like the first applications of early AI technologies to the protein world with his work at ESM, at MedAI and FAIR. to now his work in state-of-the-art contributions to structure and design. Nick extending that tradition into some of the most exciting results in ESM3, now at EvolutionaryScale, not just an evolution, but really a revolution of many of those technologies. Patrick's work across the tree of life and genomics, and now what Molly is working on, which is really the bleeding edge of how we take all of these models and apply them to develop true scientific intelligence or even superintelligence. So we couldn't put together a better topic to answer the following, and first question, and anyone who'd like to start, please do. The term foundation model comes from work that is, we know in LLMs and NLP and in vision, but its application in areas of chemistry and biology and genomics is maybe imperfect, right? There's lots of differences in the way that we think about chemistry and biology and proteomics, where that term, it's not exactly clear what that means. We have a wider set of modalities and use cases, and the end state of success and what that means and how you measure what a foundation model is less clear. So in the context of science and biology, how do you define a foundation model? What's its essential characteristics? How does it differ from the way that we think about foundation models in these other fields that we're all intuitively familiar with through ChatGPT and other services today? So, yeah.
I mean, maybe I'll try and say a few words. First, really great to be here and very excited about that. But for defining a foundation model, I think it's interesting to think about these as distributional learners. And I think we see that in sort of natural language where models that are sort of trained on all the sentences humans have uttered and stuff. It's just an incredibly rich and interesting set. And I think some of the original insights for the ESM that really motivated the early days, and again, Josh, I'm curious your perspective on it. I think we're really thinking that there was a similar distributional hypothesis for what are all the protein sequences that have ever existed in nature? And if we just understood that set and what it meant to sort of be part of that set, that really would be an interesting foundation for building application layers on top of it or other more specialized tasks on top of it. So I think for me, there really does have to be some hypothesis there. And maybe I would draw a contrast actually to some of the things in chemistry where I think that term has been a little different, and the sort of the sets of molecules that can be constructed are a little different. And I don't know, it could be something that we touch on. But I don't know, Josh, I'm curious. You were there at the very beginning.
Yeah, well, it wasn't. We didn't call it foundation models back then. I think the terms have kind of evolved over time. I think, if I remember correctly, there was a paper from Stanford a couple of years ago where they kind of put forward this term of foundation model. But before it was just a model. People called it pre-training. We would then fine-tune a model, and I think that turned into the foundation model is something you could then kind of post-train or fine-tune for different applications. But what's been super interesting to observe is actually a lot of these models these days, you're not even fine-tuning it on different applications. Like just the singular model just works across a whole host of different applications, which is really exciting. I think touching on that distributional lens, this is one of the early hypotheses, especially in biology. And I think one of the reasons why foundation models work so well for biology is that story of evolution. So if you pre-train a model on all that data that's kind of available on the internet related to DNA and proteins and so forth, you learn that story of evolution. That's where most of these sequences came from. You have scientists everywhere who are just like picking up specks of dirt and sequencing the DNA in there. And when you show this to the models, they start to learn a really informed picture of biology. I think what you'll start to see, I guess to maybe posit where some of this stuff will go in the years ahead, is there's stuff that actually isn't learned in evolution. For example, what does it mean to make a good drug? Right. Evolution doesn't care that your drug is going to be manufacturable. And those are kinds of things where I think we'll see new data sets kind of coming online, and that foundation model picture really graduating from a lot of the pre-training we're seeing right now in foundation models to biology to really connecting that with the post-training picture, and then making these models, really taking them to the next level of their utility for important things like drug discovery.
I think if you look across all the different models folks are training in the field, there's some attempt to always make your new model more foundational than everyone else's model. Some are foundation models, some are foundational models. I think they're all kind of the same, right? And I think for me, it's just a model that can be generalist, right? That's not task specific, that isn't specialized only for codons or proteins or for protein structure prediction or what have you, but something that can look across different layers of biological meaning and different length scales of abstraction, right? And I think, to Josh's point, that's what's powerful about evolution. We have a unifying theory for biology, where physicists have been trying to sort of find unifying theory for over a century now. We have this unifying force that creates these sort of natural selection and functional effects across ecosystems to individuals to organs to cells to molecules, right? Kind of across many different length scales from planetary scale all the way down to individual angstroms. And I think that's something that's really powerful, that these sort of distributional learners are able to pull out from sequence to structure to function, not just for proteins, where last year we had Nobel Prizes for protein design and for protein structure prediction, but looking across and beyond biology to the foundational information layer of biology, which is DNA, or to the dynamic information of cells and how they're perturbed in disease, right? And so I am much more interested in how these can be used for applications, right? What is the app store for biology that you can build on top of these, and how these things can actually be useful for mechanistic discovery, for drug discovery, for development, and so on. If you think about the way that we actually use these models today, they're basically sequence in, sequence out, right? And so maybe the right intuition for this is, you can use natural language and interpret what you're putting in, and you can read what's coming out. But with DNA, right, we speak DNA with a really heavy accent, right? So imagine if you were using ChatGPT, but in Russian, right? And 1% of the words were in English, right? That's the vibe of what it feels like to use these models today. And you have to build, all these crazy ways of thinking through what you're feeding into the model and what's actually coming out. And I think, trying to figure out how the models that are learning some language that we don't actually speak ourselves is, I think, sort of some of the central research questions that we're grappling with the field, in order to connect them to natural language, to how you can actually closed loop iterate on reasoning.
I think one of the things to think about as well, just as there have all of these similar underlying principles of learning generalizable features of biology, chemistry, whatever it is, there's also a lot of distinction between the different models, not also in the utility. So based on kind of what kind of underlying structure we can learn, for example, evolution, things that have an evolutionary underpinning, we've been able to start to see bigger impact on the outcome faster. I think this is also where chemistry gets a little bit harder, and why some of the foundation models that we've seen in chemistry just haven't necessarily yielded the results that we've seen in biology, and especially in proteins. And so kind of understanding where are these types of models going to be most effective. And for those that don't have that natural kind of underlying evolutionary or other type of structure that's already in built in, how do we start to build those into foundation models as well? I think that's one of a big question. I'm sure you guys thought a lot about that when building Evo as well, of how do you make sure that you get the right structure in those models?
Yeah. I mean,
important things.
Yeah, absolutely. And I mean, I think that's consistent with our experience at NVIDIA around sort of the successes of some of the work in small molecule chemistry, and versus those in these areas where the application space and the distribution that you're learning is a little bit more clear.
Right.
I remember a few years ago, somebody said something like, well, but chemistry in a foundation model is not differentiable, right? So you have to learn other things on top of it.
Right.
And so that's pretty interesting. One of the other things that I think a lot of people are working on right now is obviously in the multimodal space. And so is there, I think what we learned in just the last 10 minutes is, the application of what you're using these models for is perhaps the most important thing driving the definition of whether or not you actually have a truly generalizable foundation model. But is there a rubric for, what data is really necessary to do that? Can people think about, when they think about all the data that's being generated, what should we build? What are the types of rubrics to understand the types of data that we need to generate to be able to train models that are true foundation models for these applications. Anyone can take a crack.
I think it's hard to put an explicit rubric on these things. A lot of it is very empirical at the end of the day.
Yeah.
So one of the, I think, black magic parts of building these models is, can you actually come up with the right evaluation sets? Yeah. The gold standard for this stuff is generally going to the lab and actually testing whether these things work in the real world. that process, if you can figure out a proxy for that on the computer, really can accelerate things. So a lot of the, I think, magic we've seen, especially things like speeding up the past 2 years or so, is figuring out proxies on the computer that actually carry over to what we can see in the lab. If you talk to folks working on foundation models even outside of biology, if you ask like what's the key ingredient to make things work, of course there's like the models and there's the compute. Data is obviously one of those key pillars, and if you can't find enough data for your task, then what do? You try to find an adjacent task where hopefully there's some transfer. So I think when we think about these rubrics, it's really hard to evaluate these things by just looking at the data alone. You really need to actually kind of build that whole system and evaluate it and build some intuition of what's going on so you can figure out how to make it faster. And anything you can do to just increase those feedback loops and just make that cycle faster of learning is, I think, ultimately how this field is going to move.
I think the different types of data from like large just, sets of data that we've seen like, Uniprot or PDB, that type of data set versus this iteration data set of like learning and like actually having the computer tell you what other types of data you need is a complete, these are different paradigms. And we're going to need to move towards the latter more and more. We've kind of, just like we only have one internet, we can't get more data from the internet, we're going to see that same thing happen in biology. And this is kind of the thesis for what we're going after with Lila, is that we need to be able to generate more data, and we need to be able to generate the right data. And if you get that data in loop where you can more quickly update the policy of like what do you need next, is going to be an important next phase, I think, of this field in general.
I think defining multimodality is also important because people, use multimodality in an analogized way, right? So, typically in Gen AI, you would mean language and, vision and audio, right? But, in the EVO series of models, what we've shown is that just by training on DNA alone, because you have regulatory DNA and non-coding RNAs and proteins all embedded within DNA, there is a kind of multimodality that emerges from the model because it can design all of these individual types of molecules and their molecular interactions. But there's also, RNA and gene expression, right? There's also images of cells and tissues. And then on top of that, there's the dynamical information. There's, how this is actually remodeling in a niche, right? How this connects to a patient set of characteristics and their EHR data. And I think you'll need the right types of paired data sets across all of these different layers. I tend to sort of frame it in my head as different length scales in biology. You have a class of these sort of all atom diffusion-y models that are trying to, kind of predict proteins and pockets and ligands and what have you. You have this other layer of models that try to connect these molecules to function, right? And that's what these sort of EVO series of models do. There's a set of biological models on top of that try to start spatially and look at H&E stains, at spatial transcriptomics data, at microscopy data, and so on. And then you're kind of missing heretofore some way of stitching them across these maybe 3 buckets of abstraction and then connecting that to a higher order level of, kind of UK Biobank population level health data, right? So these are kind of the core frontier questions for how do we actually build really good models in each of these buckets or bins, and then how do we connect across the layers of fact?
Yeah, absolutely.
Yeah. I love that demarcation, and I think it gets to, you hear people now using terms like virtual cells, virtual patients, even that are
digital
designed,
twins.
that are designed to bridge. But I think that it definitely makes sense to build great models within each of these levels, and maybe to what Molly said about there are some of these very, very large data sets that exist there, and then we will ultimately need to generate specific data as well. But I do think there's an intermediate stage that we're in now, which really is about trying to connect all the heterogeneous data that has already been gathered on. This is maybe a little kind of what you said, Joshua. You want to look at that adjacent data and see can you learn things. I think I sort of have this thought for biology that we want to be in a world with our models where they are, maybe it's multimodal, maybe it's just saying they're expressive enough that pretty much any data that we believe is like high quality and has information content in it can be put inside this model so that there is the possibility for transfer. And I think that sort of paradigm has proved very effective in the more general space, and you see that with language and image models that have been combined, and I think that's sort of maybe how we overcome the kind of the heterogeneity of biology is really being able to incorporate it together.
I think it's a really interesting place to jump to a question that I had that I wanted to start with Molly is, let's say you have all of this interconnectivity and therefore a true understanding and representation of biology as you defined it. So there's lots of words for this. Virtual cell was brought up in the previous conversation. So we have that, but you're building something that is attempting to really have a scientific intelligence, a scientific agent that automates the process of doing science. This is clearly a key component of that.
100%.
What's the gap between that and really turning that into something like an automated scientific agent?
Yeah, this kind of gets back to the questions around the types of data we need as well. When we started looking at foundation models in biology, we started in places that like the human brain just really hasn't been evolved to understand. There are protein scientists that can look at a protein and they can kind of understand how it's supposed to be engineered, but our brains weren't meant to do that. Our brains are meant to like reason about like scientific principles and how you like process and the ways in which we kind of connect all this information together at a higher order. And so what we saw kind of this super intelligence in one way in protein design, protein folding. Like, yes, computers can be better than humans at that, of course. Like, it would be crazy to think that humans should be better than computers at that. But the question that we have is, what about this reasoning that's set on top of that? Just as every one of these foundation models, is learning those languages of biology, how scientists use them really matters. Mm-hmm. One of the things that I've experienced a lot is that you can have the same model of biology, give it to 3 scientists, and you get 3 completely different results. And that's because experts that understand how these models were built, how they use them, can actually use them in some really kind of expert way that isn't captured in the model itself. And so if you can get that layer of reasoning about how do you actually use these models, think about them as like just like any other tool you have in the lab. Just as you would go in and pipette liquid, you could go, understand some underlying structure of biology. And so that's kind of the other layer of learning what a scientist thinks about. And these are things that aren't written down. If you actually look in scientific literature, everybody knows this, the reproducibility problem in science is massive. And that reproducibility problem stems from the fact that how you actually do science is not well documented. Like one of the biggest advantages in some of the in the labs that many of us have been associated with is just that kind of implicit knowledge that scientists pass down from generation to generation. And we're trying to now figure out how do you actually encode that into a model. And so these are the types of places we're going. Yeah.
I just want to add to this. I think one of the reasons why, DeepSeek became so popular as a model was that it had this open box reasoning trace where you could actually see it think in real time. And it actually turned out this is incredibly interesting, right? Because you can see it doubt itself, right. Trace its steps back, kind of argue with itself internally, and then kind of, I actually found it more valuable just to read these reasoning traces than the actual summary output that it gave me at the end, right? And, the thing is, in an electronic lab notebook or a paper notebook with the, kind of messy, disorganized files that you have in the sort of process of doing biological research. You have some facts data, some spreadsheets, some NGS FASTQ files, and there some on your server, some on your Dropbox, some are on the unnetworked thing in the hallway, right? Like, you compress all of that into these, ELNs, but it's sort of like the doctor's notes at the end of a long shift, you're just kind of putting down the most bare minimum thing. The vast majority of the reasoning trace has been lost, right?
100%.
And how can you actually recover that at high fidelity in order to actually enable these agents, right? I think another way to think about it is, okay, if you have GPT, 6 or 7 or 8, right? Is that going to be the massive model that can do all the things, or is it going to be able to call a bunch of, kind of smaller, more focused task-specific models? And that's basically what folks are trying to do with these multi-agent frameworks today. And, folks are, I think Lila and others are at the frontier of exploring how you'll actually deploy them in the lab to do useful things, right? There's the Google Code Scientist. Microsoft is doing a bunch of interesting things here. And we're very interested at ARC at being at the frontier of deploying them in scientific research to actually do useful things. So we recently released this thing that we call the Virtual Cell Atlas, which is the world's largest data set of single cells. And to do this, what we did is we actually created an agent. It doesn't tire, it doesn't complain, right? And all it does is it searches the NCBI sequence read archive, the very largest database of biological data in the community, and then just finds all of the RNA-seq data and then reanalyzes it, not going from count tables, but going all the way back up to the raw data, and then aggregating the 400 million or so odd cells that are actually just the corpus of what's been created by the research community, and then uniformly analyzing that and then delivering it, right? That's the kind of thing that a human would never do, but an agent can just crank on this 24/7, right?
One thing that's so interesting here is I think these models are graduating from tools into products where they can actually own more of these end-to-end workflows. I think you just look at computational biology, computational biophysics, these things have been around for a long time, but if you look at how people were using these tools 20 years ago versus what it looks like today, there's almost like less scientific intuition that has to go into this, because we're getting closer to models now that are trained end-to-end to solve entire biological tasks. So instead of a scientist having to look at a structure prediction and then think about how am I going to go modify this protein or this molecule in order to maybe make this drug better, you instead just put it into the model and say, make me a better drug. And the model is starting to make a lot of these decisions on its own. I think it's still an open question of what is going to be the value of the scientist in that loop as well, right? We have, I guess, folks like automating the scientists now, right? And then you actually have the models just owning a lot of that decision making on its own. You see the same thing in NLP as well, right? Like calling the tools. If you look at how we did code generation even 2 years ago, the first version, GPT-3, people were even just like asking for help writing their code. You would take something there and copy it into your code editor. Now you've got things like Cursor and Codium and Cognition just doing it all for you. And we're a little bit behind, I think, in biology in terms of this really hitting us. But I think it's like fascinating to think about how the whole end-to-end workflow of solving these biology problems will change when the models actually have their agency to make some of these decisions on their own. And I think they're going to be actually way smarter than most of us as scientists.
Do you worry that we'll get dumber? Right? Like if you've used Cursor, right, you're just doing tab autocomplete. You're not really dealing with the syntax yourself. I was trying to write something the other day with O1 Pro, and it was just reasoning and then reasoning and then reasoning, and I couldn't keep writing. I had to stop and go get some coffee and then refresh it when it was like crashing. And I
definitely think
interfering with us.
I definitely worry. That's one worry, but the kind of related worry is just how are we going to train the future generation of scientists? What is that going to look like?
We'll just do what the model tells them.
Of course, yeah. Well, it turns out, this is something that we think a lot about is who are going to be the best actually users of these systems, and how is that going to change over time? There have been studies that have kind of put these types of tools in scientists' hands. And what some of the results have shown is that people who are actually already experts in the field are able to get a higher lift from these models. I think this is true in coding as well, but this has been shown in science. Actually, in material science is where this was shown. And then the junior scientists actually got less efficient because they weren't able to actually judge the output of these models well. And so what happens when those junior scientists don't become the senior scientists that we need to really be interacting with the system? So I think there's a whole question around how do we train scientists in the future that, the community will have to wrestle with.
The thing with discoveries is by definition, they're like net new, right? And so they would be judged by a model to be very unlikely, right, in terms of its likelihoods.
Right.
And so I feel like there's some way of trying to use model hallucinations in order to try to kind of generate and sort through net new crazy hypotheses and actually use that as a platform for discovery. Because otherwise I worry that we'll kind of maybe raise the floor for telling people how to analyze their Western blot, or you should actually do flow and not a blot. But at the kind of the top end of discovery, maybe we need to make sure that we're still able to do that really well.
Yeah. Right. I think if I look forward, maybe we will see the models increasingly become artifacts of study in and of themselves. It's sort of, maybe yes, there's this version of, the virtual cell or the digital twin that you can just ask the question and it can just give the answer. But I think there's probably going to be a period of time where you can prompt it and actually how the model is internally arriving at that answer is going to be of interest. And there is, this is sort of getting at things like mechanistic interpretability that I think are showing a lot of promise right now in the natural language space. And I think, it's gonna go that way, I think, in biology too. I mean, we see people doing this now in the sort of ESM family models of proteins, trying to look inside the internals of the model and actually maybe discover new biology from how the models are computing things. And so I think, maybe this next generation of scientists, maybe they're going to spend more time doing that. And, there is an aspect of the scientific method even in doing experimentations and things there that might be actually just much more productive and higher throughput than some of the things that had to be done in the lab a few years ago.
Yeah. Well, I wanted to move on from this discussion because one of the second goal of this panel was really for people to leave with an understanding of where are their success stories today, like to ground. I mean, we've talked a lot about some very high level topics here in foundation models in biology, but I was hoping to ask each of you, what's a really concrete, specific success story that you've seen? Every single one of the people on the stage here today has published in this specifically and the outcomes of the work that they're doing. So yeah, cite a specific problem and how it was solved using a foundation modeling approach. And maybe I'll start with you on the end, Joshua.
Yeah, of course. It's interesting just thinking about Patrick's point about what's net new for the models. There's so many possible inputs that you could put in here that there's not enough compute in the world to navigate that entire search space. So some of this is how are you even using the model, or what are you prompting with? To illustrate an example, there's a story I was really inspired by. There's a group of scientists that were studying this molecule where many patients would get resistance to it. And they had a lot of trouble actually figuring out the 3D structure of this molecule together with the protein. So they've been going back and forth, changing the molecule, changing the protein, putting together a picture, been working on this for years. And then they went and they typed this information into Chai1, one of our models. And then, the scientist writes in, I fell off my chair. I finally have an understanding of what's going on with this system. And I think it's an example of something where, the model might not have figured that on its own. It's something where there was really some partnership of the scientist together with the model, understanding what's going on. A few weeks later, that scientist wrote in, we use those insights. We launched another battery of experiments. Things are back on track. We're learning a lot here. And ultimately, I hope this leads to some value for patients at the end of the day, too. But I think it just goes to show that the way, at least today, there is still a lot of alpha on how do you use the models effectively. When we see this every day at Chai in our company, most of our team has been working in AI in drug discovery companies. Many folks are running AI groups in drug discovery companies. And the intuition that comes with that, how do you actually use these models? What are they capable of? Where are they headed? Actually gives you some taste and what are the right ways to be applying the models today. I think over time that might start to change, honestly, as the models do more and more. There is a question of, again, what is going to be that role of the scientist in the loop. But I think today, actually, a lot of the success stories are people being really creative on how do we actually leverage these tools and how do we actually build these tools to solve real hard problems that just can't be solved with the biological tools that are available to us today.
Talk through one of the success stories that we published in Science earlier in the year from the EvolutionaryScale team in ESM3, which was designing a novel fluorescent protein. And this was a project that just was I don't know, very near and dear to my heart. Fluorescent proteins I find incredibly exciting. These are proteins that make corals glow in the oceans, and they've been a workhorse tool of molecular biologists for maybe 30 years now, discovered much long before that. The question we sent out is if we've built one of these protein foundation models that could reason over sequence, structure, and function, but could it actually design a novel functional protein that was unlike the ones that exist in nature? And so we set our sights on something that if you shone light on it, it would glow green, because you really have to get a lot right to solve that problem. It's not just something that you can kind of like throw something out and it's going to fold up and do it. It really has to almost be perfect. And so we set a really in terms of like a prompt for the model, and I think there is a lot of art in still, like how do you prompt these models, and then use the model to design and optimize a sort of a trajectory really to find this model. And we kind of ended up with one that, from a sequence standpoint, really looks nothing like anything found in nature. We kind of estimated that it would have taken maybe, I think it was 500 million years of evolution to get from the nearest known thing to this green one. And then we put it in the lab actually with Patrick, and we saw that it glowed. And I think that was just really inspiring for me, of like, wow, okay, these models can actually they can do this in the real world. And that's my example.
I think many proteins also have accessory factors. They could be other protein cofactors, and sometimes those are RNAs, right? And so in our EVO1 paper, we showed that in CRISPR gene editing, you have a Cas9 nuclease, which is a molecular scissors, and it has a non-coding RNA guide, right? And they have to bind together to create the active complex that can go and cut DNA. And so the state-of-the-art Cas9 is actually still, one of the first ones that was sort of developed for genome editing in human cells. It's still the best that we have today over a decade later, which is sort of strange and interesting, right? But one of the things that we used EVO to do was to actually design a guide RNA that was made for Cas9, right? And today, you find these guide RNAs by mining them, literally, from genome sequencing databases. But it turns out that the sequences that you get from nature aren't necessarily evolutionarily or functionally optimal, right, for the activity of cutting DNA. And so actually, because the model can co-design a protein sequence, which is embedded in DNA, and a non-coding RNA sequence also embedded in DNA, it can co-design them, and it could actually improve the activity of the Streptococcus pyogenes Cas9, or it could design this for Evo Cas9s, which was, we thought, really conceptually novel and striking compared to many of the natural mining efforts that have occupied some of the very best labs in the field. With EVO2, we show that it's state-of-the-art in predicting the pathogenicity of disease-causing mutations, like the famous BRCA1 gene that can cause breast or ovarian cancer, right? You have mutations that can cause breast cancer, and you might elect in that case for a double mastectomy, or you have a benign mutation, but most mutations are known as variants of unknown significance, which is fancy doctor speak for we don't know what the hell is going on, right? And so, that turns out the model has an opinion about whether or not that unknown mutation in your BRCA1 gene is going to cause disease or not. And, it's able to do that simply by looking across the expanse of observable evolutionary sequences without any supervised annotation or labeling or fine-tuning or, even seeing human genomic variation, which we thought was pretty deep and powerful.
Yeah, so I'll be excited to talk about some of the successes we're seeing in really being able to automate the scientific method. But to the point of like success, especially in the field of drug discovery, oftentimes people don't really believe it until you actually start seeing something in humans. So let me tell about a success story that's a little bit older, but is foundational in that regard. And this is a molecule that we designed using some of these foundation models, these generative models, at Generate Biomedicines, where we were trying to create a drug that could be have better drug-like properties and pharmacological properties to improve patient experience. So for many patients, these drugs, especially for severe asthma, are administered in the hospital. So you actually have to go to the hospital to administer the drug that helps this population of patients. And the dosing was such that they had to go every month. And as you can imagine, that's incredibly difficult for many people to do, if not incredibly inconvenient. So we showed that we could actually design a better molecule using AI that would make them only have to go twice a year. And this was a huge advance, but it's something that we couldn't do with existing technologies. So everything to that date, as far as creating better molecules, better proteins, better antibodies, was empirical. So you had to have an assay that you could actually read out, learn from that assay and select better molecules. But the binding, the level of binding that we needed to reach for that was so tight that even the assays that we could do empirically couldn't read out the level of binding that we needed. And so we had to do a small number of experiments and design in silico these better molecules. And so by doing that, using these foundation models, we now have a molecule in phase 2 clinical trials that is proving out that hypothesis. And so we're going to continue to see more and more of these models push into the clinic. And I think as that happens, the excitement from the community will continue to go back into the research that's being done, and just driving that force, I think is going to be an essential part of getting the power out of these models.
I mean, this transitions beautifully into a question that I wanted to ask, which perhaps you've already answered, which is how do we bridge sort of the translation between in silico and vivo success and that translation gap into the real world and some of those hurdles. I mean, is there anything you'd like to add there in terms of yes, we can feed all of this back, but what is that? How do we motivate the development in a way that will lead to those outcomes?
I mean, I think this is an essential question. We get asked this often. Where all of the data is right now is in these early discovery stages. I mean, there is data in later stages. It's just hard to get access to. That's another conversation. But the more that we can pull some of these insights from the successes that we see in the clinic into these early models, we're not just speeding up the discovery stage. We're actually improving the probability of success of later stages of drug development, and that's something that I think sometimes gets missed. And also, it's just not been proven out yet because clinical trials just take time. As we start to show that we can increase the probability of success of clinical trials using some of these models, I think that's when people are going to start to really kind of see, yes, this is translatable. We're not just looking at cool biology, understanding that this underlying structure of proteins. We're actually doing something that's impacting people's lives and impacting patients. And I think the more that we're able to pull those insights into our early stage models, the better off we're going to be. So we're constantly thinking about this and trying to figure out how to do that better.
Yeah. Just to add to that, the therapeutic probability of success that Molly mentions is 10%.
Yeah.
So, from preclinical to approved drug, we have a 90%
sometimes lower
failure rate, or lower, right?
Yeah.
And so that means really two things. We're really bad at making drugs in the sense that maybe we're drugging the wrong target, or the drug composition is not doing it in the right way. And that might be a target engagement, efficiency, selectivity, PK type thing, right? And I think the vision of what we can do with virtual cell models is to accelerate our ability to find the right target or set of targets, and then also to be able to make more effective compositions. And in both cases, make this search problem, which has been guess and check to date, something that can be predictive and model guided, right? And that would hopefully accelerate the pace of discovery and then kind of over time lead to increased POS in the clinic.
Very much. I just agree with all of that. Even then getting it to the right patients as well. That's another part of this. So it's really the whole thing, and you have to have hits on all of it for it to work.
Yeah, and getting enough shots on goal as well. That's one of the other exciting things here, just the diversity of kind of molecular matter that you can come up with here.
Fire more rockets.
Allows you to, exactly. Fly more rockets, good way to put it. Yeah, or try more drugs.
So, thank you so much. I mean, I have a few different directions we could take the last few minutes that we have, and I do want to leave 5 or so minutes at the end for questions. But I think one thing that's likely on everybody's mind is that building these models is pretty expensive in general, and so I wonder if you guys could comment, coming from not only a diversity of backgrounds, in your current roles, but in a variety of different roles in each of your cases and types of organizations, the business model for these biological foundation models. Yeah, I mean, I'll leave that mostly as an open question, but I'm really curious about are we looking at a future landscape that is dominated by the real utility of these things in large pharma and biotech? What does the monetization or lack thereof look like in biotechs that you all represent today? Just curious to get your thoughts on the future of that ecosystem.
I'll say something. It's yes, training these models is expensive, but running clinical trials that fail is also very, very expensive too. And so I think when one thinks about this, there has to be this question of how good are these models, and what is the level of intelligence that they have, and how much is the acceleration there, and we touched on things like probability of success. How much does that change? And so I think there is a lot right now that is on the sort of the research side, and I think there's incredible progress there. But I think it's sort of as we get to this list of problems that we kind of enumerated, I think seeing changes there. I mean, that's I really think where the impact is going to be.
Yeah, the flip side of that too is in the tech world we have Moore's law. In the biotech world, we have Ahrams' law. Moore's law spelled backwards. The costs are going up over time. So if there's a way to place your bets on NVIDIA driving down the cost as opposed to, biology costs going down, I'm happy to put my chips on NVIDIA. So even if it costs a little bit more to do this stuff in the short term right now because of all those compute costs, machine learning scientists aren't cheap these days, all of that stuff, I actually think that that's where you want to be on the cost curve because things are going down quite rapidly there. And that ultimately should allow us to actually take many more shots on goal. The other flip side of that, which is, I think, interesting to look at too, is actually even on the lab side, a lot of the data generation stuff that's important for these models is also getting commoditized. So if you look at the kinds of experiments that we're doing today at Chai versus the kinds of experiments like we would do in our last jobs, the cycle time of lab experimentation has come down dramatically. The cost has come down dramatically. What we need to insource versus what we can outsource has also changed a lot. So we can actually just reduce our capex and fixed costs, and we can kind of move that outside. You see this even on the tech layer, right? We can build on top of public clouds instead of actually having to build up your own clusters. Obviously, NVIDIA has been fantastic partners in that for the whole ecosystem. So I think overall we're in a really exciting again part of this curve where costs are going down. It's becoming easier to build this stuff. So the companies, this question about where does even value accrue come out of the winners here? The companies that have gotten themselves positioned for this era, I think are ultimately going to be the ones that can end up winning.
So, I mean, my last question before I'm going to ask a very open-ended question to end the session that we have is, this is a deeply R&D focused community and with a lot of contributions from open science and open source and all the things that happen in the academic world as well. So, how do you think about in your own organizations the need for balancing collaboration with the open community, contributions to that community, and obviously then the need to monetize on top of ultimately what you build with them? How do you think about your own contributions to the open community, and that has enabled a lot of the R&D that's possible today?
I mean, I think this is a really important point. We spent a lot of time at Generate asking this question. It's one of the reasons that we decided to share what we were doing with Chroma and publish it. A couple points. One is that by sharing it, we've gotten a lot of feedback on the model. We've gotten a lot of input on how it's being used, how it can improve. We've improved it ourselves. And then the ability to let talent also be able to share what they're doing with the world. This is just an important part of this community. I think that's a foundational piece of who we are as scientists. And so there's a ton of value of figuring out what are the types of things that you can share that is really just, like rising tide rise all boats kind of situation versus the places that you really do need to kind of monetize in the way that you're talking about. And I think that getting that piece right is always the challenge. But sharing this ability for us to collaborate and learn from each other is an essential part of the ecosystem.
Yeah, I mean,
Go ahead.
Yeah. I think we very much agree with that at EvolutionaryScale as well. And we've put our models out. Some models we've made open source for academic usage. We also have a place, Forge, where our models can be used openly by the community. We've put models on BioNemo and other platforms, and I think it is something that's incredibly important. I think a lot of us, as you said, we come from science and research backgrounds, and I think there is just a value that comes from an academic and research community sort of pushing a field and forward. And I think it's still early in the space of foundation models for biology, and so I think, and there are so many applications that can be built on top of it, that having deep engagements with research communities, research partners is just essential. And then there are ways to think about, okay, what are commercial strategies, viable business models, size of model that gets sort of held back. There's a lot that can be done here. So I think it's not really like an all or nothing. I think.
Right.
Yeah, I mean, I think it's there's a number of commercial advantages to also kind of putting your work out there with the community. When we started Chai, we knew the opportunity here was so grand that there was no way we were going to be able to do everything ourselves. So we knew that partnering with the community was going to be important. And a first step towards that, we open sourced for free our first model for companies, for academics. And there are a couple of reasons for that. I mean, first of all, we're in the early innings of this stuff. The models are going to get a lot better. There's also going to be some level of commoditization that happens with the models, and you just have to be aware of that. And if you know it's going to happen, like, why not get ahead of that curve? And I think it also then leads on a commercial strategy to building a lot of trust in that ecosystem as well. If you're going to be a partner with someone, how do you actually prove that you're going to be reliable, that your systems are going to work, that you're going to be best in class? And the most scalable way to do that is to actually just kind of prove it out with the community. So we could go hire teams of chemists and biologists internally to go and evaluate the models at scale. And we do a little bit of that, but it does not compare to the scale of what's happened in the open source community with our models. And just seeing the kinds of use cases that people are exploring, people are writing in with like the kinds of examples that they're seeing. We have just so much that we've just learned by doing that exercise that I think it really just propels those future efforts as well. So I think finding that right balance in your portfolio of like what you're going to publish, what you're going to partner, what you're going to do yourself is really important to get right.
Yeah. I think people talk a lot about open versus closed source in the sort of foundation model game for natural language, for example, right? And the thing that you see in the revenue between OpenAI or Claude or X or Grok or Mistral or what have you is, there's this sort of ecosystem lock-in based on, how you build your agent workflows on what API call and, which, I think people just use the tooling that's built on top of these models, not just the sort of, weights and the parameters themselves, right? And so I think, when we released Evo 2, right, with NVIDIA, one of the things that we worked very hard to do was to build an Evo designer so that folks could actually use the tool and feed DNA in and read it out and interpret it and fold the proteins embedded inside of the generations, to build a visualizer for all of the mech-interp work that we did when we trained SAEs on the model to find feature activations. How can you actually look across them and actually match them at the nucleotide resolution to all the genome annotations, right? These are the things that allow people to actually use these things, because otherwise you're just dealing with things that are kind of foreign and confusing. And I think that's really where you can draw, adoption of your tools, right? These things become useful when other people use them, not when they're Nature and Science papers alone.
Honestly, I think that's very consistent with our experience at NVIDIA as well. Although we're not, operating and doing the biology, we rely on feedback from the community and the tooling that we're building. And so through our collaborations with many of you on the stage, I mean, that's part of our philosophy as well. I mean, we are still doing research here, right? And so without feedback from the people that are using the tooling that we're building, whether that's solving a biological problem or solving a computer science problem, we probably can't make very much progress moving forward. Okay, so we have like 1 minute left for like the last question that I wanted to ask. And so perhaps I'll ask you to be very concise in this. And I'm gonna piggyback on something that Patrick began this whole conversation with earlier, just an hour ago, which was, what's the app store? And so what I'm going to ask is, what's the killer app of foundation models in biology 10 years down the road? What is something that we're going to be really, really excited about? What is something that's going to surprise us about the effectiveness or the utility or the outcomes of all the work that's going to happen over the next 5 to 10 years in this space?
Can I just say science?
Sure.
I think that's like the biggest question in my mind, and the theme has been throughout the entire conversation. That is, how do you start to connect all of this together? At the end of the day, like one of the biggest challenges is that every model is disconnected from the other, not just from each model, but how you actually use them.
Yeah.
And so that might sound like a little bit of a cop-out, but I actually don't know that any one application is going to prove out the power of foundation models in biology. Like, at the end of the day, if there was only one application, like, we wouldn't be investing.
Yeah.
At the rate we're investing. And that's kind of the thesis of where we're headed, is that as you start to actually connect these fields together, connect these models together, connect how you actually use these models, that's going to be the killer app on top of it. And then you're just going to have many applications that are going to have value in the field. I don't know.
I think you could count it by biopharma % of GDP. How much does that go up, right? But
Yeah,
I think
right.
drugs are just a small part of biology. Biology can terraform planets. It can make biomaterials. It can be used for computation, right? I think we should think bigger.
Yeah, maybe it's sort of programmable biology, just being able to really engineer it in like a rational, intentional way. Like, okay, I want this little nano machine that's going to do this, and maybe this is something therapeutic, but maybe it's that, and we can just treat it like like an engineering discipline in that way. Awesome.
Yeah, I don't think anyone's going to tell Anthony the real killer apps because there's a lot of alpha on that, right? But I think broadly we're going to have maybe two main axes. There's things that everyone is doing in the lab today, and you'll probably just see AI incorporated everywhere, right? Like things we're doing today, we'll be able to do it faster, cheaper, better. But then we'll also have AI just enable like totally new categories that we just aren't in reach today, and I think that's incredibly exciting. I think there'll be a lot of surprises along the way too, as we start to stress test these methods and see what we can learn from them.
Brain-machine interfaces, so we merge with AI.
Well, thank you all so much. I hope you're all inspired. I certainly am. That's why I like to end these panels with some open-ended questions like that. We have basically 3 minutes for maybe 1 or 2 questions at most. So Rory's up here with a mic that he can pass around if anyone wants to raise their hand to ask a question. We'd certainly love to answer it. And yeah.
Thank you for a lovely panel. So as we're thinking about the uplift that these foundation models can provide for therapeutic development, how do we think about their less desirable uses? For example, creating not therapies, but perhaps the opposite of that. And how do we think about controlling that, if we can at all, at the digital level and then perhaps at the physical level?
It's a great question. Anyone want to?
I mean, I think this is something we think about a lot. And there's like a couple different ways to look at it. One is that how do you control people from intentionally doing bad things? And then how do you control people from unintentionally doing bad things? And they kind of take two different realms, and we work a lot on that. The kind of unintentional bad things, that like the harmful things that could happen, that's existed in science, as long as we've been able to synthesize DNA, essentially, and maybe even before. And so we have some standard kind of biological safety mechanisms built into our labs. I'm sure every lab, up here has that. I think the more interesting question is, how do we start to control like people from intentionally doing bad things, or once we allow like more autonomy in these labs, making sure that the LLMs themselves aren't doing something that's not aligned with what human scientists want done. And so that's like a bigger open research question in our mind, and some place where we're spending a lot of time thinking about what are the actual scenarios that we need to be considering, what are the safety levels we need to be considering, how do we think about how we're reaching those safety levels in a similar way to how frontier models are thinking about it, just with another layer of scientific labs built in. So as we start to advance some of those safety frontiers, just in the frontier models, well, we're going to have to bring that into biology as well.
Yeah, absolutely. I think this, you sort of think about this in context of really responsible development, and there are a number of things that people can take all the way from thinking about what training data is going into models. Both Patrick and our team have encountered that in terms of excluding potentially infectious agents from training data, and also think about how models can be prompted. And so I think it's very important. I mean, we'll say biology. It is the space where the ability to do things like design things for harm, and the ability to design things like rapidly develop antidotes. These things can go hand in hand in some ways, and so just really being intentional on that along the way is, I think, really important.
Awesome. Well, thank you very much for the question. That's all we have time for. I hope you'll join me in thanking this incredible panel. They had a great time.
Thank you, everyone, for joining. I think the only wrap-up is you guys need to go to the back to take questions and get unmiked. This will be online for any of those that want to tune in afterwards. Thank you.